Namaste students, I am Jayoti Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth, Women's University, Jaipur. Today I am going to discuss about the topic biological control of the microbial growth. In the previous video, I discussed about the control of the microorganism growth by use of the some physical agents, heat, and some disinfectant, chemical disinfectant. Now I'm going to discuss about the control of the microbial growth by use of and some biological agents like antibiotics. So what these are also known as the chemotherapeutic agents, and they are the chemical agents used in treatment of many diseases. So in this series, and biological agent is antibiotic. You know that antibiotics are biological agents. They are Antibiotics are the proteins which are secreted by the microorganisms to protect themselves against some another microorganism. So each microorganism which have capacity to produce antibiotics, they can protect themselves uh, from another microorganism because these antibiotics are the protein molecule. They are toxic molecule which can kill another microorganism. So we can use these antibiotics for the protection of the human also. Okay. So for that we need to produce these antibiotics in the fermentation methods, and we can produce these microbial antibiotics by growing these microorganisms in a suitable culture medium. So antibiotics are special type of the chemotherapeutic agent which are used in treating infections, disease, and they are usually obtained from the living microorganisms. If we talk about the history of the antibiotics, European in 1630, they used natural quinine of the silicon natrium. It was used for the treatment of the malaria. In 1910, Silverasan was used for the syphilis. Okay, and sulfonamides were also used in 1935, and sulfadizine and sulfamadizine was used in 1945, and then in 1920 the significant discovery of a miracle that was happened, that was the penicillin. The penicillin was discovered by the Alexander Fleming in 1929, and the drug was the first out in the market in 1950s. And what is the mechanism of the action of the antibiotics? How these antibiotics can kill microorganisms? So here we are going to discuss about the mechanism. First thing is that these antibiotics will inhibit the cell wall synthesis in the microorganism. How the beta-lactam, penicillin, cephalosporin, and carbapenem, and monobacterials are the antibiotics which inhibit the growth, which inhibit the synthesis of the cell wall. Next, the protein synthesis inhibition. Not only the cell wall, but also the protein of the microorganism are also inhibited for the synthesis by these antibiotics. Okay. The binding of 15 ribosomal subunit, which is exhibited by the macrolides and the chlorothalonil and the cyanidamine, or to the 30S. So these antibiotics do not allow to binding of the 50S subunit to the 30S subunit. So if the small unit of the ribosome will not bind to the large unit, that is the 50S unit, then the protein synthesis cannot be formed. The protein cannot be synthesized until 50S unit and 30S unit will bind to each other. some antibiotics inhibit the binding of the 50S amino, and some antibiotics like amino glycosides and tetracycline inhibit the binding of the 30S amino to the 50S amino. So it is not bound that the protein cannot be synthesized. Next, third number mechanism is that these antibiotic interference with the nucleic acid synthesis also. Okay, like RNA by inhibition of the RNA by rapamycin and inhibition of the DNA synthesis by the fluoroquinolones. Next is the inhibition of metabolic pathways like sulfonamides inhibit the metabolic pathway. Next is the disruption of the bacterial membrane structure. Some of the antibiotics can disrupt the bacterial cell membrane, like polymyxins and daptomycin. So these antibiotics will inhibit the cell membrane synthesis, and they will disrupt the cell membrane of the microorganism. Now we need to talk about the different properties of the good antibiotic. What are the different properties of a good antibiotics? First is the selective toxicity. The antibiotics have some selective toxicity. They are harmful to microorganisms, but without being harmful to the host, they are not harmful to the host. Like if we are using these antibiotics for ourselves, so we need to inject, we need to take it in a pill, and that antibiotic will not be harmful for us. It will be harmful only for the microorganisms. It will not be harmful for humans. Okay? And it must not be harmful for the humans. So that must be a property. Second thing, the antibiotic must be broad spectrum. It must be of broad spectrum, and it inhibits many different species of the pathogenic microorganisms. Third thing, it must prevent development of genetically and phenotypically resistant strain. So no more strain, no more resistant strain should be developed. It must be unique, and no resistant species must be generated by these antibiotics. Next thing is that these antibiotics must have bactericidal activity. Means they must kill the bacteria rather than stop the growth of the bacteria. So they must kill the bacteria. Fifth thing is that it must not allergic and no adverse reaction. It must not show any adverse reactions. Sixth number, the antibiotics should remain active in presence of the plasma and other body fluids. Seventh is it must be stable and water soluble. The example include the streptomycin, cefixime, tetracycline. So these all are the examples of the antibiotics. The different groups include the penicillin, cephalosporins, quinolones, sulfonamides, aminoglycosides, tetracycline, chloramphenicol, some antifungals like ketoconazole, fluconazole, itraconazole. So these are the examples of the antifungal agent also. We can also discuss about the antibiotic uh, structure. So different antibiotics may show different structure. They have a particular structure. It means whatever they have a particular ring structure. They have a particular carbon structure. Okay. Uh, what is the antibiotic resistance? Antibiotic resistance is the ability of a single bacteria to resist any antibiotic. Okay. to resist an antibiotic. Means that and the microorganism can survive when we give any antibiotic to those microorganisms. So they will not die. So they develop a resistance against a particular antibiotic, that is known as the antibiotic resistance. So what is the need of the drug resistance? Drug resistance is important in prohibition of the resistance in healthcare institution and treatment failure which causes serious consequences. What is the reason of the antibiotic resistance? Antibiotic resistance is due to the over prescription of the antibiotics and non-completion of the prescribed antibiotics. It is due to the use of the antibiotics in animals as growth enhancers, and it is due to the poor hospital hygiene. It may be due to the indiscriminate use of the antibiotic. Means if we are using more and more antibiotics, then sometimes the microorganisms which are present in our human body, they will develop resistance. They will get mutant and they will get resistance. And after that, there will not be any kind of effect of those antibiotics on the microorganism. What is the mechanism of resistance to antibiotics? First is the upregulating the production of the enzymes that inhibit the antimicrobial. Means we upregulate the production of some enzymes. Okay. Then they may develop the resistance of the antibiotics. Similarly, upregulating the pumps that expel the drug from the cell. So these are the mechanisms by the antibiotic resistance can be developed. How the antimicrobial activity can be developed in vitro drug pathogen resistance is very important. First thing is that the environment of drug and pathogen is very important. The environment influences the effect of the microorganism. Microorganisms are located in different tissues and parts of the body, and the antimicrobial activity will have important role, an important effect on a particular organism, on a particular organ, on a particular part of the body. Different parts will show different activities. So environment is very important. Next, the state of the metabolic activity is also important. A diverse biosynthetic activity in the microorganism is also important. The distribution of drugs, how the drug is equally distributed among the tissues and the body fluids is also important. Next, the location of the organism is also important. The drugs enter the cell at different rates. So, if the location of a particular and distribution of the drugs, how the drugs is distributed into different cells is also important. And what is the interfering substances which are present into the host is also important. Sometimes some drugs can interfere, some substances can interfere with the drugs. Okay. And sometimes the drug can also react with the blood and tissue proteins. Okay. So if that is also very turning point in the next the concentration of drug is also important. Organism inside the body may not be exposed to that constant concentration of the drugs due to some factors like absorption, distribution, variability of the concentration, post antibiotic effect. These are the factors. And the host pathogen relationship is very important because alteration in tissue response is important. Is dependent on alteration in the tissue response is also dependent on the effect of drug. And alteration in the immune response is also dependent. because sometimes the immune response of the individual can also change. Sometimes it is lower, sometimes it is high. It depends on the individual to individuals. And similarly, the alteration of the microbial flora is also important, because microbial flora can sometimes increase in number, and they can also increase. They can also change their behaviors. Like they can be commensals, but they can convert into the more pathogenic forms. Post-drug relationship is also important. Like what are the effect of the absorption, metabolism, what are the distribution of the drug, is the drug is toxic to the humans, or is the drug is excreted out of the. And now we discuss about the different dangers of the antibiotics. What are the different dangers of antibiotics? So, widespread sanitization, changes in normal flora of the body can be found, and similarly, masking that it can also result in the masking of the serious infections. It can also result in the direct drug toxicity, and also result in the development of drug resistance in the microbial population. So, these all are the effects. We can also test the antimicrobial susceptibility by the minimal inhibitory concentration and minimal bacterial concentration detection, and we can also detect the antimicrobial by the dilution method, two-fold dilution method, or Agar serial dilution method. So these are the dilution method by which we can analyze. Similarly, we can also analyze the effect of antimicrobial agent by the disk diffusion methods. Okay, like we can dip our antibiotic into the uh, kiwi- into the agar within by the using the kiwi bore technique. We diffuse our drug, and we also inoculate our culture medium. If there is a zone of inhibition around the disc, then it will be found that the disc is effective against the microorganisms. So these are the methods by which we can analyze whether our drug is effective or not. Similarly, the another and biological agent which is used for the uh, treatment of the microorganisms, vaccine. Vaccines are the some microorganisms, that the microorganisms which are attenuated, which are inactivated. Okay? So the microorganisms are either attenuated or they are inactivated in different forms, and they are injected to the human body. So the after injection into the human body, the body will develop some immune, it will show some immune response against the attenuated or the inactivated microorganisms which are inserted in the form of the vaccines into the body. So body will respond. Okay? Body will respond, and it will generate some antibiotics. It is considered as a primary immune response. It is known as a primary immune response, and the primary immune response will take a longer time to respond. You know that. And when the same type of microorganism will enter into the body originally, then the body will not take a longer time. It will respond immediately because the body has developed some memory cells inside it, and these memory cells will remember the microorganism in the lifelong. Okay? So these vaccines will protect the body in the whole life until there is not any mutation in the microorganism. So this vaccine, the vaccine is a biological agent which is inserted inside the body, and this biological agent is the microorganism itself, and the microorganism is attenuated and it is inactivated. Okay, so the micro for the microorganism, the immunity will develop, and this immunity will protect the human in the whole life. So these are the two examples which I discussed in this video lecture. So I hope the students have better understood all these things. If you have any query, if you have any questions, so you can ask me so anytime. The question which can arise from this lecture is that explain the mode of action of antibiotics, how the antibiotics can protect. The second question which can arise, uh, explain the role of the biological agent in controlling the microorganism growth. So you can answer in two ways: the antibiotics and the vaccines. So two forms for the protection. So my lecture is over here. If you have any query, if you have any answer questions, so you can ask me in the YouTube channel and comment. So my lecture is over here. Thank you very much.